Hi class, this is your instructor, Skylar Huff. We are now in Chapter 26, Viruses. We'll begin our exploration of the diversity of life with viruses. Viruses may seem like an unusual place to start, as they lack many of the characteristics of life. On the other hand, their ubiquitous association with all organisms, their impact on those organisms, and their relative genetic, biochemical, and structural simplicity make them a good place to begin. At a bare minimum, all viruses contain genetic material enclosed in a protein shell. Despite their relative simplicity, their disease-producing potential and ecological impact make viruses important biological entities. The virus particles pictured here are recreated from genomes of the influenza virus responsible for the 1918 influenza pandemic, which killed an estimated 50 to 100 million people worldwide. Other viruses cause diseases such as AIDS and the common cold, and some even cause certain types of cancers. So as we go through this lecture here, we'll spend a great deal of time on the nature of viruses, a little bit of time on the diversity of viruses, and then we'll get into the bacterial viruses, and then viral diseases of humans, and then end things with prions and viroids. So early virologists made important discoveries in molecular genetics, and their work gave us important tools used in biotechnology and genetic engineering. So with the discovery of giant viruses, newly emerging viral diseases, and advances in viral ecology due to modern genomics, virology remains one of the most exciting fields in modern microbiology. All viruses have the same basic structure, a genome of RNA or DNA surrounded by a protein shell called a capsid. If the capsid is tightly associated with the genome, then it is called a nucleocapsid. Capsids and nucleocapsids are typically composed of one to a few different protein molecules that assemble together to form predictable shapes such as icosahedrons or helical filaments. Some viruses have an envelope of lipids, proteins, and glycoproteins derived from the cells they infect, and others have more complex structures. Viral particles, virions, are not cells because they lack metabolic activity and do not have cytoplasm and cannot replicate outside of a host organism. Viral genomes can be linear or circular and single or double-stranded. Viral genomes are highly diverse and serve as one way to classify viruses as discussed in the next section. Without a suitable host, a virus cannot independently replicate its genome or make the proteins required to assemble new virions. Also, as evolution requires transmission of genetic material, viral populations cannot undergo evolution without a suitable host. When viruses infect cells, however, they usurp normal cellular processes and acquire some of the characteristics of living things. Viruses have found suitable hosts in fungi, prokaryotes, and protists, as well as in the cells of plants and animals. Scientists have found viruses in all organisms where they have looked for them. Particular kinds of viruses can infect only particular types of cells. Either a virus can recognize only one kind of cell type, or it cannot replicate efficiency in a cell type. The cell or organism types that a particular virus can infect are referred to as its host range. Within a multicellular host, many viruses exhibit tissue tropism, targeting only a specific subset of cells. For example, rhabdovirus, the virus that causes rabies, infects and replicates only within neurons. This tissue tropism will determine the nature of the infection and the disease symptoms caused by any particular virus. The primary function of a virus is to make more copies of itself. This function is encoded in the viral genome. Although viral genomes include genes for making capsid proteins, they often do not code for proteins involved in the expression and replication of their genomes, and never contain genes to make ribosomes. This means that viruses have to take over the host cell's machinery to replicate their genomes, express their genes, and use the proteins produced to build new virions. In some cases, a virus must carry with it protein needed very early in genome replication or expression. This is the case because some viral genomes are very different from their host cell's genome. For example, HIV, which infects specific immune cells in humans, carries an enzyme called reverse transcriptase, which makes DNA from an RNA template. Reverse transcriptase must be carried by the virus because the single-stranded RNA genome of HIV must be converted to double-stranded linear DNA upon infection. There is no equivalent host enzyme, so the virus must provide this enzyme for early events in infection. Once inside a cell, a virus hijacks the transcription and translation machinery to make new virus. There are three general types of viral genes expressed during infection: early genes, which contribute to taking over the host cell's expression machinery; intermediate genes; and late genes, which are usually capsid proteins and proteins involved in viral release from the infected cell. Although an infected cell might produce thousands of new viral particles, this does not always kill the cell, and the cell can become a virus-producing factory. Most viral capsids are either helical or icosahedral, although there is significant variation in these two basic structures. Helical capsids, such as that of the tobacco mosaic virus, shown to you here, have a rod-like or thread-like appearance. Icosahedral capsids, such as those of the adenovirus, shown here, appear generally spherical. Their icosahedral geometry is revealed only when viewed with an electron microscope. The icosahedron is a structure with 20 equilateral triangular facets. Most animal virus capsids are icosahedral. The icosahedron is the most efficient symmetrical arrangement of subunits to form a shell with the maximum internal capacity. There is an upper limit on how large viral genomes can become if accompanied by an icosahedral capsid, and this may be the limiting factor in viral complexity. Some unusual strategies to accommodate larger genomes have been adopted by some viruses, and those viruses, such as the pox viruses, have more complex structures. While capsid structure is relatively well-defined, overall virus morphology is highly diverse. Viruses with no envelope, called naked viruses, will either be filamentous or icosahedral, like the poliovirus shown in figure 26.2, shown to you here. So it's a naked virus with an icosahedral capsid. The capsid is formed from multiple copies of 4 different proteins, and this image is a digitally generated 3D model. Some bacterial viruses, such as the T-even bacteriophage shown in figure 26.3, are complex. Complex viruses have a binal or two-fold symmetry that is neither entirely icosahedral nor entirely helical. The T-even phage in figure 26.3 has an elongated icosahedral head connected to a hollow tube with a helical symmetry ending in a complex base plate with tail fibers. Envelope viruses have much greater morphological diversity than naked viruses due to the flexible nature of their envelope. Many envelope viruses, such as influenza viruses, are generally spherical, but, as can be seen in the photo at the beginning of the chapter, are somewhat variable in shape. Some envelope viruses, such as the bullet-shaped rabies virus, have more defined and uniform shapes due to the viral capsid that provides the structure to the envelope. So here what you're seeing is an example of a complex virus. So at figure A, shown to your left, it's an electron micrograph, and figure B is that typical structure. So this is T4, the bacteriophage. So what you're seeing here is viruses as they vary in shape, and if you look closely, there are dramatic differences in the sizes of cells and viruses and the diversity of viral morphologies. So you see the pox virus near that yeast cell, and the bacterium there to the top left. Herpes simplex virus is shown, rabies virus is shown, HIV is shown, thereafter influenza, the adenovirus, the poliovirus, we're getting smaller and smaller, the West Nile virus, and then you have the protein hemoglobin, and then here at the bottom you have the T2 bacteriophage, and then you have the bacterium E. coli. Viral genomes are either DNA or RNA, linear or circular, and single or double-stranded. Approximately 70% of all viruses have RNA genomes. This includes the viruses that cause influenza, measles, and COVID-19. RNA genomes can be single or double-stranded, with one debatable exception: are linear. Because most host cells cannot make RNA from an RNA template, viral RNA genome replication uses a virally encoded RNA-dependent RNA polymerase. This RNA polymerase is highly error-prone, which leads to high rates of genome mutation. This makes viruses with RNA genomes difficult targets for the host immune system, vaccines, and antiviral drugs. Although many RNA genomes encode genes on a single molecule of RNA, some medically important viruses, such as the influenza viruses, have segmented genomes. These genomes have several pieces of RNA, each with one to a few genes. The retroviruses, which includes HIV, have single-stranded RNA genomes that are reverse transcribed into double-stranded DNA by the enzyme reverse transcriptase. In HIV, double-stranded DNA version of the genome is integrated into the host cell's chromosome DNA, where it is expressed to make viral proteins and new viral genomes. Other viruses, such as the viruses causing smallpox and herpes simplex, have DNA genomes. Most DNA genomes are double-stranded and usually replicated in the nucleus. So you have important human viral diseases shown here, just as you see in Table 26.1 in your textbook. So there is chickenpox, a varicella zoster stranded virus. No cure, and it's rarely fatal. Vaccines have been available since 1995. And a higher dose of the 1995 vaccine is available to protect against shingles. We have hepatitis B, the viral form. Herpes from the herpes simplex viruses. They are very, very prevalent worldwide with no cure, and exhibits a latency, and the disease can be dormant for several years. Then there is mononucleosis, causing Epstein-Barr virus, or at least caused by the Epstein-Barr virus, spread through the bodily fluids, especially saliva. We have the cervical and penile cancers caused by the human papilloma viruses. There is a safe and effective vaccine available for HPV 16/18. Then there is AIDS caused by HIV. In 2013, the World Health Organization estimated that 35 million people are living with AIDS, with an estimated 2.1 million new HIV infections and an estimated 1.5 million deaths. The Zika virus, the West Nile fever caused by the Flava virus. We have Ebola. Peak mortality is 50 to 90% if untreated. There is influenza, and vaccines have to be remade each year, and they are available. There is measles, caused by the viruses, extremely contagious. Vaccines are available for it, and usually it is contracted in childhood. When it is not serious, it is more dangerous to adults. We have SARS, and then the SARS-CoV-2, rabies from the rhabdovirus. Commonly infected animals include bats, foxes, skunks, raccoons. Giant viruses. Viruses are significantly smaller than cells, having diameters from 20 to 250 nanometers. Virus, originally misidentified as a bacterium that infected amoebas, was subsequently found to be a virus of 750 nanometers in diameter with a 1.2 megabase or 1 million base genome. The identification of several additional giant viruses followed the discovery of virus. Pandoravirus, isolated in 2013, has a micrometer-sized virions and a 2.5 megabase genome, which is larger than those of some bacteria. The largest currently known virus is pithevirus, which was revived from 30,000-year-old Siberian ice in 2014. With virions 1.5 micrometers long, pithevirus is larger than some bacteria and even some eukaryotic cells. Metagenomics, the study of genetic material isolated from the environment and not directly from organisms, has been used to identify a new group of giant viruses, the Viruses. The role of these giant viruses in ecosystems and disease biology is currently unknown. If size alone is not enough to challenge assumptions about the distinction between viruses and cells, the genomes of some of these giant viruses also contain features not previously observed in viral genomes. For example, before the discovery of the giant viruses, no viral genome encoded proteins involved in translation. The genomes of isolated giant viruses collectively possess 9 of the 20 aminoacetyl tRNA synthase enzymes, and the hypothetical Clostridium genome contains genes for all 20. The presence of parts of the translation machinery in the giant viruses suggested the hypothesis that they represented the 4th domain in the tree of life, sharing a cellular ancestor with the other domains. However, phylogenetic analyses of the gene sequences of the aminoacetyl tRNA synthase genes reveal that the giant viruses were not descendant from a cellular ancestor. Rather, the evidence suggests they acquired the translation genes from hosts as once smaller viruses. Despite the fact that all viruses are obligate intracellular parasites, viruses show significant diversity. This diversity is the basis for which several different virus classification schemes. Viruses are most commonly classified based on taxonomy, the type of disease the virus causes, the host range or tissue tropism of the virus, or genome type and expression. Different kinds of classification systems meet the needs of different kinds of scientists. A medical microbiologist might be most interested in a classification based on disease type, while molecular geneticists might find more utility in the classification based on genome expression. The use of metagenomics to identify novel genomes like that of Clostridium make it increasingly important that viruses are classified and can be referenced consistently. The choice of classification system should consider whether it allows predictions about the group members given the characteristics of the group. So they can be classified by way of taxonomy. They can be classified on disease, based on disease or host. Henceforth, there are 4 types of influenza virus: influenza A, influenza B, influenza C, and influenza D. And the different types can cause varying degrees of symptoms or no symptoms at all in animals as diverse as primates, pigs, poultry, bats, cats, dogs, and cows. They can be classified based on genome expression. And genomics is driving advances in viral ecology. So I won't get into each of those, but I would say classification by disease or host. So both of these methods are limited, and not all viruses cause disease. Some viruses cause different diseases under different conditions or times during infection, and some viruses, like the common cold, can be caused by several viruses. And then lastly, some viruses infect different organisms, i.e. influenza. So given what we have here in figure 26.5, this classification is based on the relationship between genome organization and the production of mRNA for gene expression. So the positives and negatives indicate the sense of the strands of nucleic acid. Plus sense strands have the same sequence as mRNA, and minus sense strands have the complement of plus sense strands. So most viruses are in prokaryotes and protists, and they're hard to study in the lab. So metagenomics lets us study all viruses. So you isolate the DNA from the environment sample, amplify and sequence and compare it to the database. So it's found thousands of new kinds of viruses. And just think, that last bullet, seawater has 10 times more viruses than bacteria, which is a big source of genetic diversity. Bacteriophage, or phage for short, are structurally and functionally diverse viruses that infect only bacteria. Most bacteriophage have a characteristic head-tail morphology that is found only in bacterial viruses. Although they are rarer, there are also bacteriophage with only icosahedral or helical capsids, which can be enveloped or naked. E. coli phage were among the first discovered and remain among the best studied viruses. One category of viruses is called the T series, T1, T2. To illustrate the diversity of the T of viruses, T3 and T7 phage are icosahedral and have short tails. In contrast, the so-called T-even phages are more complex, with an icosahedral neck and a neck with a collar and whiskers for a long tail and a complex base plate. Viral infection usually results in the production of new virus particles, which, when released, may kill the host cell. Released virions can infect other cells, perpetuating an infectious cycle. This kind of infectious cycle, called a lytic cycle, because the virus usually causes the cells to rupture or lyse. Some bacterial viruses, called temperate viruses, can also enter a lysogenic replicative state, which involves the incorporation of the genome into the host cell's genome. These genomes do not produce virions, do not kill the host cell, and are transmitted from cell to cell when the cell divides. Certain triggers can cause a lysogenic infection to become a lytic one, with the resulting death of the host cell. The lytic cycle of virus reproduction is illustrated in figure 26.6, and the basic steps of the phage lytic cycle is similar to those of a naked animal virus. Although the basic outline of this infective cycle is common to most viruses, the details vary, as do the viruses themselves. First is attachment, or adsorption. The virus contracts and becomes specifically bound to the cell. This step limits both the host range and the tissue tropism of viruses, as viral surface proteins will only bind up to specific proteins on the surface of host cells. The second step is penetration. The virus introduces its genome into the host cell through the bacterial cell wall. This has been studied in detail in phage with head-tail morphology, such as bacteriophage T4. Once contact is established, the tail sheath contracts and the tail tube passes through an opening that appears in the base plate, piercing the bacterial outer membrane and then the cell wall. The viral genome is passed through the contracted tail into the cytoplasm of the host cell. Once inside the host cell, the viral genome takes over the cell's replication and protein synthesis machinery in order to synthesize viral structural components. These are then assembled, the assembly phase, to produce mature virus particles. In the release phase, mature virus particles are released either through the action of virally encoded enzymes that lyse the host cell or by budding through the host cell wall. This shows that, the lytic cycle there on the left. Attachment, penetration, synthesis, assembly, and really. One form of lysogeny occurs when a temperate phage infects a host cell. Rather than entering that lytic cycle, its genome is integrated into the genome of the host cell. This integration allows the virus to be replicated along with the host cell's DNA, the integrated phage genome, called the prophage, and the cell containing the phage, called the lysogen. In a lysogen, the expression of the prophage genome is repressed. Although the lysogen is quite stable, it can be induced to enter the lytic cycle, if you look here to the right, from integration, upon attachment and penetration, integration, propagation, some cell stress, and then induction. If the cell is stressed, the prophage can be de-repressed, expressing the enzymes necessary for excision of the genome, and the lytic cycle can then commence. The switch from lysogeny to lytic cycle is called induction because it requires turning on, inducing the genes necessary for the lytic cycle. During lysogeny, viral genes may be expressed from the prophage at the same time as the host cell genes. When the phenotype or characteristic of the lysogenic bacterium are altered by gene expression from the prophage, this change is called phage conversion. One example of phage conversion occurs during all lysogenic infections. The host cell is resistant to the subsequent infection by the phage of the same type. Because many infections are lytic, this affords an obvious selective advantage on the host cell. Due to the number of bacterial species that can be lysogenized, lysogeny likely plays an important role in bacterial evolution. Sometimes phage conversion can change an otherwise benign bacteria into a highly pathogenic strain. Vibrio cholerae is usually a relatively benign curved bacterium found living in water. However, disease-causing strains are also known. If ingested, these strains cause the disease cholera. Each year, cholera causes acute diarrhea in about 3 million people, killing approximately 120,000. Cholera is transmitted mainly via fecally contaminated drinking water and is therefore most dangerous to the 2 billion people who lack access to clean water. An infection and cause disease, a virus must first have access to tissues in the body. Although skin is exposed and therefore seems to be the most vulnerable to infection, the most common point of infection is respiratory epithelia. Skin is a protective barrier to infection because it has multiple layers. The outermost layer, which is keratinized and is slightly acidic. Viruses can breach the skin barrier, where it is broken by injury, via feeding insects, and by the damage caused during medical procedures. In addition to lacking the protective features of skin, respiratory epithelia have a surface area about 70 times that of skin and are exposed to a continuous flow of potentially virus-containing air as you breathe. Viruses can also gain entry to the body at the gastrointestinal mucosa, the surface of the eye, and the urogenital epithelia. Once a virus has infected a cell at the site of injury, it must replicate in that cell and may cause disease either there or after it is spread to secondary sites. There are 2 main mechanisms by which a virus can spread: in blood or lymph via neurological tissue. In blood or lymph, viruses may be associated with immunological cells or be freely circulating, a condition known as viremia. Viral infections causing disease can be categorized as acute or persistent based on how rapidly and frequently the virus is produced and on the appearance of associated symptoms. Persistent infections can be latent or chronic. However, there is variation in these classifications. Acute infections involve replication of virus, which can lead to the sudden onset of symptoms. Either these infections are controlled by the immune system and resolve, or they cause the death of the host. Although there are exceptions, acute infections are often established at the site of infection and are non-life-threatening. Given the amount of virus produced, the site of production, and the typical symptoms, virus-causing acute infections are easily spread in the air and are commonly responsible for outbreaks, epidemics, and pandemics. So this shows, as you're seeing here, acute in the case of influenza viruses or the rhinovirus. They're at A. Next, along the continuum, you have chronic, such as in the case of hepatitis or even HIV. Latent, such as in the case of the herpes viruses. And lastly, those which are slowly progressing, such as cancer-causing viruses or prions. Influenza in humans is caused mainly by either influenza A or influenza B, with influenza A more clinically important. These are viruses you are vaccinated against if you get the seasonal flu vaccine. Influenza viruses are enveloped, type 5 viruses. They spread via direct transmission of spectrum of sputum in coughs and sneezes through inhalation of aerosol virions or by contact with a virus-contaminated surface. Influenza virions attach to a specific surface carbohydrate on respiratory epithelial cells using a viral envelope protein called the hemagglutinin A. The virus is then endocytized into the host cell's cytoplasm. The 8 segments of the viral genome are released from the capsid and transported into the nucleus. The viral genome is replicated and used to make mRNA, from which viral proteins are then translated. Viral capsids containing the genome segments are assembled at the cell membrane and the virion buds from the cell surface. The viral envelope is created from the host cell membrane. Release of the virus from the cell surface depends on a viral protein embedded in the host cell membrane called neuraminidase. Disease is partly due to the tissue damage caused by the virus itself, but mainly due to the inflammatory response of the host. Hemagglutinin as well as neuraminidase in the viral envelope are the 2 main viral proteins most commonly recognized by the immune system. As inflammatory cells of the immune system respond to the virus, infected cells are killed, and this contributes to tissue damage. While extremely unpleasant, most cases resolve without an intervention. However, in patients with weakened immune systems, disease can be severe, leading to respiratory failure and death. Ironically, a strong immunological response can be as dangerous as a weak one. The strength of immune responses triggered by the 1918 influenza A virus may explain the high mortality rate during that pandemic. Replication of the single-stranded segmented RNA influenza genome uses a viral RNA-dependent RNA polymerase. This enzyme makes approximately 1 mutational area per replicated genome. The high mutation rate leads to antigenic drift in the HANNA genes, referring to the hemagglutinin as well as the neuraminidase genes. Antigenic drift refers to the small changes in the HANNA proteins that the previous vaccine-induced immunity has no longer protected. Because the antigenic drift is due to random mutations, it is impossible to predict precisely which viral strains will emerge each year. Immunologists and epidemiologists at the Centers for Disease Control and Prevention use information on circulating strains and epidemiological data to make the best possible predictions for which strains to use in next year's flu vaccines. When you receive a flu vaccination, you are protected against certain strains of influenza virus. The vaccine primes your immune system to recognize influenza viruses with certain HANNA proteins. If you are infected with a strain similar to the one you are vaccinated against, your immune system will quickly recognize and control the virus. If you are unfortunate enough to become infected against an influenza virus that has different variants than those of those proteins, you will not be protected, and the disease will run its course as your immune system mounts a slower response. Excuse me. While antigenic drift causes minor changes in the HANA proteins, a process called antigenic shift can cause major changes. Antigenic shift happens only in influenza A viruses because only they have the ability to infect a wide range of animal hosts. Antigenic shift occurs when an individual is infected with more than one virus and the viral genomes are reassorted during infection. Antigenic shift producing new HANA combinations was responsible for the three major flu pandemics of the 20th century: the Spanish flu of 1918, H1N1 killed 50 to 100 million people worldwide, the Asian flu of 1957, which is H2N2, killed over 100,000 Americans, and the Hong Kong flu of 1968, H3N2, infected 50 million people in the United States with 70,000 deaths. The first pandemic of the 21st century started in February of 2009, and by mid-May, 11,000 people in 41 countries have been affected. Modern genomics provides us a unique view on how the virus causing this pandemic arose. In the 1900s, three influenza viruses infected pigs: a classical swine flu virus, a North American avian influenza virus, and a human H3N2 virus. When all three viruses infected the same cells, the 8 genomic segments from each virus are all replicated. When new viruses are assembled, the 8 genomic segments are selected randomly, which can shuffle the genomes from different viruses to produce new combinations of genome segments. Because HA and NA genes are on different genome segments, this can produce viruses with new combinations of HA-NA proteins. Such a recombined virus circulated in swine herds in North America, but could not be transmitted to humans. Sometime later, pigs harboring the new virus were infected by an avian-like influenza virus. This allowed a second reassortment in the pigs to create a new virus. This virus, called H1N109, more commonly swine flu, could now infect humans and cause disease. This reassortment created the virus with an HA protein much better suited to binding to the human respiratory epithelia. Coronaviruses are enveloped viruses with plus-sense single-stranded RNA genomes. And this is figure 26.8, particularly looking at the bottom with 26.8b for the swine flu of 2009, which I remember. So as I said, they're enveloped viruses with a plus single-stranded RNA genome. 3 coronaviruses cause moderate to severe disease in humans: MERS-CoV, SARS-CoV, and SARS-CoV-2. Coronaviruses are zoonotic. They can infect a variety of non-human animals, and occasionally they spill over from their non-human hosts into humans, where they can spread rapidly to cause epidemics or pandemics. There are 7 coronaviruses that infect humans, 4 of which cause relatively mild cold-like symptoms. Of the other 3 coronaviruses that cause disease in humans, SARS-CoV and SARS-CoV-2 are the most important. SARS-CoV first appeared in southern China in late 2002 and spread to create an epidemic in about 24 countries before being contained in 2003, which I remember hearing about this when I was younger. No cases of SARS have been reported since 2004. SARS-CoV causes severe respiratory disease characterized by fever, general flu-like symptoms, and possibly a dry cough. Many of those infected developed pneumonia. The virus is spread by close contact, most likely via respiratory droplets expelled during coughing or sneezing, but can also be spread when objects contaminated by aerosolic droplets from an infected person are touched by the uninfected. SARS-CoV-2 was identified as a causative agent of the coronavirus disease in 2019, called COVID-19, in the Wuhan province of China in January of 2020. The virus spread globally, causing about 1 million infections in the 3 months from January to March of 2020. By June of 2020, the number of infections globally reached 10 million, causing about 500,000 deaths. By mid-November 2020, the World Health Organization was reporting about 58 million infections and nearly 1.4 million deaths globally. The Centers for Disease Control and Prevention was reporting 12 million cases of infection and over 250,000 deaths in the United States alone. Towards the end of 2020, pharmaceutical companies in the United States, Europe, and Russia began to report vaccine providing 70 to 95% protection against infection. SARS-CoV-2 infects respiratory epithelia by binding via its spike proteins to the ACE2 protein found on the surface of cells in the respiratory tract. Once inside the cell, the viral genome is then replicated and expressed, and new virions are assembled. New infectious virions are released from the cell by exocytosis and can infect nearby cells, spreading the infection. Individuals infected with SARS-CoV-2 exhibit a range of morbidities. Many are asymptomatic, but others develop systemic symptoms such as chills, fever, fatigue, myalgia, and malaise. Others exhibit a range of severities of respiratory tract manifestations: sinusitis, rhinitis, cough, and chest pain. In severe cases, the disease progresses to cause acute respiratory distress, pneumonia, kidney failure, and death. Some individuals develop neurological symptoms such as dizziness and headache, and in some cases, there are digestive complaints. In individuals that clear the infection and recover in 2 to 3 weeks, there can be long-term complications from infection, heart palpitations, carditis, impaired lung function, hair loss, and skin rashes, and certain psychiatric manifestations such as depression and anxiety. Understanding COVID-19 is made more difficult because of highly variable infection to fatality ratio. And that is the relationship between how many people get infected and how many die from the disease as a function of age. The IFR, the infection to fatality ratio, is lowest for children aged 5 to 9 years old and increases linearly from about 0.05 for children 10 to 14 years old to over 1.0 for adults 65 and over. There's also significant variation in the IFR when considering other demographic variables, such as by country and by biological sex. So this is showing you SARS-CoV infection and the replication mechanism. So you have those ACE2 receptors, and then you have those spike proteins that bind to the ACE2 receptor on the cell surface, which then invites in, by way of endocytosis, SARS-CoV-2 or COVID-19. In persistent viral infections, the virus or its genome can be detected in the host for long periods of time, usually years. In many cases, the persistence is lifelong. Based on the amount of virus present and when it's produced, these infections can be classified as chronic, latent, or slow. These classifications are complicated by how and when symptoms manifest. We will use human immunodeficiency virus, the virus causing AIDS, and herpes virus infections to illustrate the chronic and latent infections. HIV infection. Well, HIV transmission occurs when contaminated body fluids come in contact with a mucous membrane or directly enter tissues. The two most common routes of HIV infection are intimate sexual contact and the sharing of needles by intravenous drug users. Like many chronic infections, the initial stages of HIV infection follow acute infection dynamics. During the acute phase, a high viral load is characterized by as many as 5 times 10 to the 6 virions per milliliter of blood plasma. At this stage, virus is spreading throughout the body, establishing infections in many tissues. The adaptive immune system is not yet responding to the infection, and antibody-based tests for infection appear negative. Symptoms of this stage, often called acute retroviral syndrome, are typically flu-like, but in many include diarrhea and joint pain. About 3 months after infection, the adaptive immune system begins to control the infection, and the number of viruses in the body drops dramatically. The immune system may control the replication of virus for many years. The low viral load during this time is characteristic of a chronic infection, and those infected are generally asymptomatic. Ultimately, however, the immune system cannot control the virus. As the viral load increases, the symptoms of AIDS appear. HIV infects and ultimately destroys certain types of immune cells. Immune cells are characterized based on cell surface proteins. The cells targeted by HIV are cells that express the antigen CD4 and are called CD4 plus cells. The most important cell types infected by HIV are the T helper cells, which regulate the immune response, and macrophages, which engulf non-self materials such as pathogenic bacteria. HIV infects and kills the CD4 plus cells until very few are left. Without these crucial immune system cells, the body cannot mount a defense against invading pathogens. AIDS patients die of infections that are usually benign. These diseases are called opportunistic infections, and they are part of the progression from being HIV infected to having AIDS. The CDC defines AIDS based on level of CD4 plus cells in blood. A person is said to have AIDS when the CD4 T cell count drops below 200 microliter of blood, or 14% of the total T cell population, and when a person has at least one of the HIV-associated diseases or syndromes. HIV is a retrovirus. It has 2 identical single-stranded plus sense RNA genomes that must be converted to double-stranded DNA by reverse transcription before being expressed and replicated. Reverse transcription, you will recall, is the use of an RNA template to make a piece of complementary DNA. Reverse transcription uses the enzyme reverse transcriptase, which is not encoded by the human genome and must be carried into cells by the virus itself. In the case of HIV, the DNA version of the genome is inserted into the host cell genome. Without this step, viral replication cannot occur. Figure 2610 summarizes the infectious cycle of HIV. Embedded in the envelope of HIV is a glycoprotein called GP120 that recognizes and binds to the CD4 receptor on target cells. Successful infection also requires either the CCR5 or the CXCR4 co-receptor on the host cell surface. Once the CD4 and a co-receptor are engaged by GP120, the viral envelope and the cell membrane are drawn close together and fuse. The 2 nucleocapsids with their enclosed genomes are spilled into the cell. Once inside the cell, the genomes are released from their nucleocapsids and converted to double-stranded DNA by reverse transcriptase. The reverse transcription is inaccurate and produces at least 1 mutation per viral genome replicated. Some estimates suggest that there is more genetic diversity in the viruses of an HIV-infected individual than all of the influenza viruses worldwide. This variation creates serious problems in the treatment of HIV infection and in the development of HIV vaccines. The double-stranded genomes are carried into the nucleus of the host cell with a viral enzyme that integrates them randomly into host chromosomes. Transcription and translation of the viral genomes produce envelope, capsid, and several other proteins involved in viral assembly. New RNA genomes are produced by transcription of the integrated viral DNA and are assembled into immature virions at the inner leaflet of the cell membrane. Interactions between the cell membrane and some other viral proteins causing the budding of virion, the viral envelope with its embedded proteins, is derived from the host cell membrane as the virus buds from the cell surface. HIV, like other enveloped viruses, does not lyse infected cells. However, when viral loads and replication rates are high, the stresses on infective T cells and macrophages can result in their death. This, in combination with virally induced programmed cell death, is responsible for the reduction in immune function that leads to the establishment of opportunistic infections that often leads to death in AIDS patients. And this is that figure called 2610, showing you the HIV infection cycle. Attachment, entry into the CD4 plus cells, replication and assembly, and then ultimately budding. Early regimes for treating HIV infection used only a single drug. Due to the high mutation rate during reverse transcription, resistance to some of the drugs appeared quickly and spread rapidly throughout HIV populations. The advent of highly active antiretroviral therapy, HAART, which used multiple drugs at once, largely overcame the problem of resistance to single drugs. In many cases, if the timing of treatment is appropriate, the depletion of CD4 plus cells late in the infection can be effectively reversed using HAART, and the disease progression can be slowed or even halted. Antiretroviral drugs target different aspects of the viral replicative cycle. Early drugs focused on blocking the process of reverse transcription. Other drugs interfere with the viral attachment, the integration of the viral genome into the host genome, and the viral assembly, maturation, and budding process. Resistance to an antiretroviral drug arises because the drug represents strong selective pressure on the virus. Mutations in the viral genome that render the drug ineffective are selected for, and these mutations will spread in the viral population. One drug, Maraviric, has the unusual property that it does not target the virus. Maraviric binds to the CCR5 receptor on the host cell and blocks attachment of the viral envelope protein GP120 so that the viral attachment and entry is blocked. Resistance to Maraviric is highly unlikely for two reasons. First, because rates of mutation in the host cell are orders of magnitude lower in the virus. The chances of a CCR5 mutation that blocks Maraviric binding is incredibly low. Second, there are no selective advantage to a cell with CCR5 mutation that interferes with Maraviric binding. Subsequently, there is no pressure selecting for that mutation in the host cell. There are two well-documented reports of people living with HIV having been cured of their HIV infections following bone marrow transplants. So one happened with Timothy Ray Brown, who was diagnosed with HIV positive in 1995. In 2007, Mr. Brown developed acute myeloid leukemia. In an attempt to treat the cancer, Mr. Brown received a bone marrow transplant from a donor who had a rare mutation in the CCR5 co-receptor that makes cells resistant to HIV, resistance to infection by HIV. Mr. Brown was later declared to have been cured of HIV despite the bone marrow transplant and his cure of HIV infection. Mr. Brown tragically died from a relapse of leukemia on September 29, 2020, aged 54. 63 other people living with HIV have been found to have been cured of the infection. However, this is due to the virus being contained in part of the host cell's genome that is not expressed. Viruses establishing persistent infections have the advantage that they can exist in the host population for long periods of time. In contrast, viruses causing acute infections may be eliminated from the population before new susceptible hosts become available. Persistent viral infections that are latent are characterized by an initial acute phase of infection followed by periods of no or low viral load and intermittent episodes of viral replication and disease. Herpes virus infections. There are 8 herpes viruses, all of which establish latent infection in humans. Herpes simplex viruses 1 and 2 are epidemic worldwide, and over 4 billion people are infected. Upon initial infection with HSV1 or HSV2, there is an acute episode of disease at the site of infection, characterized by the formation of painful blisters. Upon infection, both viruses replicate rapidly in cells at the site of infection. Infection evokes an innate immune response, which in itself causes some tissue damage. After this initial response, an adaptive immune response is triggered that resolves the infection. During the time the immune response takes to resolve the disease, virus can migrate from the site of infection to the endings of sensory nerves innervating the infective tissue. The virus runs along the axon of an infected neuron to the body of the cell, where the virus establishes a latent infection in the nucleus of the neuron. HSV1, which is primarily caused orofacial herpes, will migrate to the trigeminal ganglia, a collection of neurons located just in front of the ear. HSV2, which is primarily responsible for genital herpes, will move to the sacral ganglia located at the base of the spine and lower back. HSV1 and HSV2 can remain hidden in their respective ganglia because the viruses do not produce many proteins and infected neurons do not trigger strong immune responses. The mechanism of viral latency remains unclear. However, it is largely controlled by a single viral mRNA and several microRNAs that suppress the viral genome expression. There is little advantage to a virus in remaining perpetually latent. HSV1 and HSV2 can leave their latent state in neurons of ganglia in response to certain environmental triggers. Suspected triggers of latent virus reactivation include changes in hormones in the body, exposures to UV light, and immune suppression. When a herpes virus is reactivated, new viruses are produced, which travel back down the neurons to the original site of infection, where the new lytic infection is established. This leads to an outbreak of disease characterized by the same but milder symptoms that were seen in the initial acute infection. Given the presence of intimate contact between humans, this is most likely when the virus will spread to a new host. Several viruses are known to contribute to cancer in humans. Some of these viruses cause cancer only in severely immunocompromised individuals, such as AIDS patients. Virally induced cancers can be caused in a variety of tissues which largely reflect the site of infection or a site to which the virus can disseminate following infection. Although there is great diversity of types of virus causing cancer, most have certain characteristics in common. First, virtually all use DNA as their genetic material at some time in their replicative cycle. Second, all have some mechanism for making sure that their genome is stably inherited during cell division. Third, all must effectively evade the immune system to establish some kind of persistent infection, given the length of time that cancers take to develop. HPV is the most common sexually transmitted infection, and certain high-risk types of HPV cause a variety of cancers in humans. Possibly the most significant of these types of cervical cancer, of which approximately 70% of cases are caused by HPV infection. While some of the HPVs that infect the anogenital area are benign and causing only wart-like growths on the skin, infections of mucosal epithelia by HPV-16 and HPV-18 can cause normal cells to behave like cancer cells. This transition is slow and usually associated with the integration of the viral genome into the host genome. Expression of all but two viral genes is suppressed. The E6 gene and the E7 gene are necessary to drive tumorigenesis. E6 and E7 are viral proteins that interfere with the normal regulation of the cell cycle and apoptosis. During a normal cell cycle, the retinoblastoma protein controls the activity of transcription factor called E2F. When the retinoblastoma protein is bound to E2F, progression of the cell cycle is prevented. When the cellular conditions are right for cell growth and division, the PRB, which is that retinoblastoma protein, which controls it, releases the E2F regardless of whether cell division should occur. This forces cell division when an infected cell is receiving signals to not divide. Fortunately, a fail-safe mechanism exists to prevent this kind of aberrant cell division. If levels of E2F become abnormally high, as they do when E7 forces release of E2F from the RB, a host cell protein called P53 triggers apoptosis. However, the virus can overcome this fail-safe mechanism. The viral E6 protein can bind to inactivate P53 and prevent it from triggering cell death. Uncontrolled cell division in the absence of apoptosis to regulate cell number is often an early step in tumorigenesis. There are effective vaccines for some of the cancer-causing types of HPV, which can significantly reduce risks of cancer due to HPV infection. An emerging disease is one that appears in a population for the first time, or whose geographic spread or incidence is increasing. A re-emerging disease is one that caused significant illness or death in the past, whose incidence is on the increase after a period of low incidence. Disease can emerge due to interactions between social, economic, and biological variables. For example, changes in sexual behavior or drug use may facilitate the spread of a virus, or an increase in global travel due to prosperity may allow viruses to access new susceptible hosts. Alternatively, genetic change, such as that seen each year in certain influenza viruses, can create strains not easily defeatable by the immune system. As humans expand into environments where animals carrying viruses harmful to humans live, there is an increased risk of transmission of viruses between non-human animals and humans. In some cases, a virus may jump a species barrier that it previously respected. For example, HIV emerged into human populations when a related virus, simian immunodeficiency virus, infected humans from a monkey host. Unfortunately, emerging and re-emerging diseases often take healthcare professionals, governments, and epidemiologists by surprise. An outbreak of hemorrhagic fever caused by the Ebola virus in 2014-2015, spreading rapidly through West Africa, becoming a pandemic before a response could be mounted to control the disease. The Ebola virus has a high mortality rate of 25 to 70%, and with approximately 20,000 infections globally, resulted in more than 10,000 deaths. Prions are proteinaceous infectious particles. So they cause transmissible spongiform encephalopathies. For instance, you've heard of likely mad cow disease or bovine spongiform encephalopathy, scrappy in sheep, and then Creutzfeldt-Jakob disease in humans. The host has normal prion proteins, and those misfolded proteins are what cause that normal to misfold and cause disease. Viroids, a bit differently here, are naked molecules of circular RNA, and they range in size from about 250 to 400 nucleotides, some of which cause severe disease in economically important crops. Viroids infect plants via breaks in the epidermis or by being injected into cells by feeding insects such as aphids. Viroid replication uses a plant cell RNA polymerase to be able to make RNA molecules from an RNA template. The polymerase makes the single RNA molecules that contain repeats of the viroid genome that self-cleave into individual viroid genomes using RNA catalytic activity specific to the RNA molecule. Mature viroids travel from cell to cell in plants via plasmodesmata. During viroid replication, small interfering RNAs may be produced. These can interfere with plant growth and development and cause mild to severe disease. So to sum up this last section here, particularly in respect to prions, they are an unusual group of diseases that are characterized by the presence of numerous small cavities in the brains of infected individuals. So these cavities are caused by the loss of neurons and can take decades to form following that initial infection by that causative agent. So these transmissible spongiform encephalopathies, they include scrappy in sheep, the bovine spongiform encephalopathy, or mad cow disease, and then chronic wasting disease in deer, yes, and Kuro and Creutzfeldt-Jakob disease in humans. So they can be transmitted experimentally, which I won't get to, but in the late 20th century, the disease Kuro discovered among members of the Four people in Papua New Guinea. Members of the Four group, primarily women and children, engaged in ritualistic consumption of the dead. This led to the spread of the commonly fatal TSE through the tribes. Because women engaged in the practice more than men, the disease resulted in a significant gender imbalance in the tribes. Mad cow disease widely among the cattle herds of England in the 1990s because cows were fed bone meal prepared from sheep and cattle carcasses to increase protein content in their diets. And like the four, the British cattle were eating tissues of animals that had died of the disease. And lastly, in the years that followed the outbreak of bovine spongiform encephalopathy, there has been a significant increase in Creutzfeldt-Jakob disease in England. Some cases appeared to be genetic. However, some patients with no family history of Creutzfeldt-Jakob were being diagnosed with the disease. This led to the discovery of a new form of Creutzfeldt-Jakob called variant Creutzfeldt-Jakob disease that is acquired from eating meat of bovine spongiform encephalopathy infected animals. So concern grew that variant CJD may be transmitted from person to person through blood products, similar to the transmission of HIV through blood and blood products. So this has been your instructor, Skylar Huff. Take care of yourselves, study well, and let me know if you have any questions. Thank you all for listening.